Hello out there in the virtual world. My name is Jordan Person. I am a nurse for Grassroots America magazine and a contributing writer. Today, we are here for another episode of Meet the Scientist, where we are bringing our magazine to life and we are introducing you to the scientists, researchers, advocates, and activists that make our publication what it is. Today, I have with me the amazing Dr. Igor Kovalchuk. He is a professor at the Department of Biological Sciences for the University of Lethbridge in Alberta, Canada. Thank you so much for being here with us today, Dr. Kovalchuk.
Good morning, afternoon, everyone. Thanks for having me.
Absolutely. So we're going to dive right into some questions so we can learn a little bit about what it is that you do, that you study, and what brought you to this path. So you study the genetic and epigenetic regulation of plant responses to stress. Can you explain exactly what that means and what brought you there?
Right, so I've been doing plant biotech for the past, I'd say 24, 25 years. My main area of research before I came to cannabis was plant response to stress. And there are different levels of response to stress. So plants are able to adapt to stress fairly efficiently and fairly quickly and pass the information to the progeny. Specifically, I give you an example. Imagine you're a company that produces seeds, right? And you want to produce seeds for, say, Alberta or I don't know, Minnesota, for example, right? And you have two sources where you produce seeds, either in Minnesota as well itself, or in Chile or New Zealand. So you want to have another cycle. And then when you've done that, when you plant it, you notice that seeds from two different sources perform absolutely differently. One is more resistant to the conditions where it's supposed to grow, and the other one is not. So that's epigenetics to you. When you propagate the same genetically identical seeds in different environments, one would be adapted to this environment and another would not. So I'm studying the effect of environment on the capacity of plants to adapt and checking what are the molecular mechanisms of it. So.
Wow, that's fascinating. How does that pertain to your work with cannabis now?
Right, I worked on cannabis since probably about 5 years. So before that, I worked on medicinal poppy for probably about 12 years now. So my passion about medicinal plants came from the fact that I have a medical degree, although I'm not a practicing doctor, excuse me, but the idea and notion that plants have all the capacity we need in terms of maybe not curing entirely ourselves, but substantially helping with different symptoms and syndromes and whatnot, made me believe that plants that are used in folk medicine, used for thousands of years, have great capacity to be very useful. And cannabis sativa, whether it's marijuana or hemp, is one of the probably most unique and versatile plants that provides you with food, feed, shelter, and medicine, right? So it can feed us and animals. It can provide us with construction material. It can provide us with clothing, with paper. And in terms of medicine, don't ask me to start. There is such a broad variety of things that cannabis does, simply because we kind of co-evolved with cannabis. So cannabis has been around as long as we have been around, and continuous history of safe consumption made us to respond to this plant very efficiently. We have our own so-called endocannabinoid system, right? So that basically it's a homeostasis system. So to dumb it up or simplify, if anything goes up in your organism, cannabinoids would bring it down. If anything goes down, they would bring it up. So they balance your response. And in many conditions or situations when either with age or with inflammation, something else, the endocannabinoid system is not sufficient to cope with that. And that's where we could substitute it with phytocannabinoids from cannabis.
Excellent. That was one of the most beautiful descriptions I've ever heard the endocannabinoid
Thank you.
system described. Yes, no, thank you. I can't wait for our viewers to listen to that description. So many things went through my mind when you were talking just now, including the research capabilities in Canada. What exactly, when you speak of studying cannabis sativa, are you allowed to study the whole plant? Are you studying a synthesized molecule, cannabinoid, or are you actually allowed to study whole plant matter?
Right, right. So yeah, in Canada, the rules are a bit is up. You still require the application to work with cannabis, but it's fairly straightforward process. It still takes probably up to a year to get a license, but I have a research license since about 4.5 years now, and right, so I can grow my own plants, I can breed them, cross them, I can order cannabinoids from other companies. I can import, export any material for research purposes to most of the countries in the world. So I can get seeds from others. I can cross them, hybridize. I can work with animals. I can prepare material for clinical trials. So virtually everything. Yeah.
Go Canada is basically what you're saying, because we are light years behind you, sir. Here in America, we are light years behind you. So wow, that's incredible that you have access and the ability to study the plant that way. That blows my mind. um so you clearly are on the forefront of the biotechnology of this plant medicine. Are you studying any other plant medicines, or is cannabis one of your primary focuses right now?
Right. So as I said, we worked on poppy. So in fact, my lab produced a special type of medicinal poppy, so-called seabane poppy. So where we genetically engineered poppy not to produce a morphine, right? So morphine is a final alkaloid, and seabane is the one that is upstream. So there are ways how to block the pathway to morphine. What it does basically, if the plant that produces morphine, it can be easily used for heroin production, right? So for illicit purposes. But if you block this pathway, sebane is a main alkaloid that is used for production of synthetic painkillers like OxyContin. You don't need morphine for that.
Right.
But the problem is for illicit purposes that you cannot make heroin out of it. So again, like our lab produced this plant a while ago, maybe 5-6 years ago. We also work on other plants, not only in cannabis, so other anti-inflammatory. We work on dandelion. We work on several tropical plants, let's say, that produce amarin and other anti-inflammatory plants. We just recently started working on psychedelic mushrooms as well. So.
I wondered, since that's coming across the world right now like wildfire, I
Yeah.
wondered if that was something that you were going to get into or already were into. And
Right, because they have very strong anti-inflammatory that perhaps rival some of the cannabinoids as well. So yeah, it makes sense to study what they can do, right?
absolutely. I have used psilocybin throughout my life for its PTSD purposes, and I had surgery. I had my clavicle hardware removed, and we were having a ceremony, and my pain completely left my shoulder. It was not something that I had ever utilized psilocybin for previously. And I mean, I had my sling on still, and I had range of motion. It was absolutely unreal, their capabilities for pain. I had never known that they had these anti-inflammatory effects. So I am so ecstatic to see the future of both medicinal mushrooms and of cannabis. The world of potential is so unreal. The nurse in me, you know, I spent years in the medical field in so many hospitals in different settings, shoving prescriptions down people's throats. And now here we have the potential of these plants is just so incredible.
Yeah, it's all severely understudied. I just wish that US would open more doors for researchers because, well, let's face it, you're number one country by capabilities of research, and not doing research on cannabis is bizarre to me because countries like Israel, Germany, they're good powerhouses, but they're not in forefront. But or Italy, to this matter, or Canada. I mean, we're probably, I don't know, 7, 8. Well, in the capability, but not number one. And we've done already so much more. So when you join, it will be a revolution. So I hope you join very soon.
I hope so too. The COVID pandemic, I feel, really woke a lot of the country up when they realized that it became an essential thing. They became essential workers, the trimmers, the growers, the sellers of the product.
Yeah,
Everyone became essential for the first
right.
time since the 1800s. You
Yeah,
know,
yeah,
when we were originally told to grow the plant in this country, and then everything changed. And
yeah.
so I agree. I hope that politically the climate changes someday soon and that we can join you in this amazing race revolution that's to come. And speaking of revolution, you recently went viral with a study that you have been doing with some other amazing scientists regarding the possibility of cannabis sativa products possibly being effective for COVID-19, possibly the respiratory distress that is a result of COVID-19, and the other symptoms that are a result of COVID-19. Can you speak a little bit about that to kind of give us a teaser in case you want to come back and tell us more?
Right, right. So obviously, we were not doing any COVID research before February, March this year, right? So, but in the past 4.5 years, we have bred about 1,500 different hybrids, crosses between marijuana, hemp, you name it. We just did it agnostically, and then we would grow the plant, take flowers, make an extract, and study variety of anti-cancer effects. We tested against 7 or 8 cancers and profiled up to 400 different extracts, identified many that are very efficient, so we've got patents on this and stuff like that. Then we also did anti-inflammatory effects, so where we would use single cells like intestine cells, epithelial skin cells, oral cavities, buccal and whatnot, and 3D tissues. So they're so-called human engineered skin or human engineered gut or human engineered lung epithelial cells. That really, it's like I'm saying it engineered, but it's exact human tissues from donors, right? And we would induce inflammation with a variety of agents, either with UV or molecularly with TNF and interferon, for example, and then apply different extracts and see which ones would decrease the inflammation cost. So when COVID started, we already had a set of data. And so we just reanalyzed, identifying the components that are targeted in COVID. Our first idea was that this virus resembles SARS virus in mode of action, and it was known that SARS targets gateway tissues through the ACE2 receptor. So it's basically a protein on the surface of the cell that has, let's say, structure that fits one of the proteins of the virus, S protein, right? So this protein can interact with the receptor, and that's basically how viruses are tissue specific. They target tissues where the receptors are the best for them to interact, right? So, and so target tissues for us is basically oral cavity, gut, and lungs. Mm-hmm. And since we've done research on those, all we did, we reanalyzed the data and identified the extracts that actually decrease the ACE2 receptor by up to 80-85%. So to give you an idea, so if you have a room, right, with 100 doors and you have 10,000 people outside that are entering these doors, they will enter at certain speed. The more doors open, the faster they enter. Now, let's just imagine, it's silly, I know, but let's imagine zombies are hunting you. And you have to enter very quickly. And zombies are our macrophages, so that eliminate the virus, right? Our immune system, right?
Mhm.
So if you close suddenly 85 out of 100 doors and only 15 is open, there would be simply not enough people enter, let's say, to start a party, right? And that's how the virus works. It needs certain titer to get to the enough cells. And if you close those doors, there's a chance that you will not be infected, right? And so that's where the idea of prevention came. So right now, we are in the midst of the study for mouse wash and gurgle that uses one of our cultivars. So the idea is that you use it either preventatively or as soon as you've been diagnosed but don't have symptoms yet, so that it reduces the load of the virus, right? So because we have a lot of immune cells in our oral cavity and our throat, so mouse wash and gurgling continuously washes off those viral particles, right? We're hoping that would have a significant effect. So that was the first study, and just yesterday we received the confirmation that has been accepted for publication because it was initially as a preprint, right?
Mm-hmm.
I cannot release the name of the journal yet because of embargo, but within 2 weeks it will be out, and then you will know about it.
Fantastic.
Yeah, so the second study was based on anti-inflammatory effects. So again, we found what interleukins, interferons, and cytokines are targeted by SARS-CoV-2. And so we profiled target gateway tissues and identified those cultivars that substantially reduce the expression of TNF, interleukin 6, interleukin 8, and again, so identified those cultivars that do that very significantly. Preprint was out about 4 months ago. We're now going through the second revision of the paper. It just demonstrates the vigor of this. So the first one, it took us 3 months of rebuttal and additional experiments to prove that yes, what we are showing is not a fluke. So the same going on about the second paper, and we're hoping that we will convince the reviewer. So that is extremely important because if the reviewers believe us through multiple rounds, that the public should believe us too.
Absolutely. Absolutely. Well, that's why, as I was researching you in general, this was the first thing that came up. And I thought, oh my goodness, yes, I actually shared that myself because I believed it immediately.
Yeah.
We have this local theory, which is just, a few of us that are in the cannabis industry, that none of us, or even a few of my friends that actually came down with COVID-19, they've all been regular cannabis consumers. And none of them have had the severity of symptoms the way that other people have that were not cannabis consumers. And so it was just our, conversation theory that there had to be something that had to do with cannabis and its multitude of capabilities, including being one of the strongest anti-inflammatory, antioxidants, neuroprotectants that exists. And
Right, right.
so thank you for doing this research. I can't wait to put your new published study all over our social media platforms. Dr. Kovalchuk, for those of you out there listening, has had over 150 peer-reviewed publications and holds over 20 patents. So thank you for taking the time out of your insanely busy schedule to spend some time with us today to tell the world what it is that you're doing. We greatly appreciate you and the advances in science that you are making for this industry, for this plant, for the world. Do you have anything that you would like to add today, Dr. Kovalchuk?
Not much, except that reciprocally, Jordan, I should thank you and people like yourself who actually doing this wonderful job spreading information, because for something as revolutionary as cannabis, that being banned for many, many years, without you and people like you that take risks, that expose yourselves to the authorities and whatnot, we wouldn't be here in Canada and hopefully one day in US, where the research will be freely available. And one day, everyone can grow their own medicine and know that it can be done safely, securely, because it has been studied. So.
Oh, thank you so much, Dr. Kovalchuk. I greatly appreciate your time.
Thank you very much. Okay, take care. Bye-bye.